Our study area for the fieldwork involves 3 places, which are Hutan Simpan Gunung Arong, Pantai Penyambung, and Pulau Batu Gajah. Then, our target population would be all the aquatic or marine invertebrates located at the study area. There are a total of 3 activities done during the fieldwork. The first activity was done on the second day, which is on the 1st December 2023 at Hutan Simpan Gunung Arong. This activity is divided into Part A and Part B. Part A involves sampling activity. So first thing first, we measure a square plot with its length and width 5 meter. Then we measure the objective factors, which are the humidity, temperature, pH level, light intensity, and water velocity. Next, we sample the aquatic insects and invertebrates by using 2 methods, which are via the server net and aquatic net. Each of the samples are placed into a plastic bag and sealed tightly. For Part B, we identify and determine the aquatic insects and invertebrates. The samples inside the sealed plastic bag were poured into a serving basin and placed into a rectangular basin. We then observe the morphology of the sample by using a microscope and then identify the organism using Google Lens and reference book. After that, the samples of the same characteristic are placed into the specimen tube and labeled accordingly. Second activity was done on the same day at Pantai Penyambung. In this activity, we observe the marine invertebrates that reside at the shore and place them together inside the plastic bags. We also make sure that we take pictures of them so that we can identify them using Google Lens and also sources from the internet. The third activity was done on the third day of the fieldwork, which is on the 2nd December 2023 at Pulau Batu Gajah. This activity involves us observing and identifying the marine invertebrates. We first swim in the shallow water at the area not far from the shore. Then, we took pictures of the invertebrates we came across there. Apart from that, observations of invertebrates that reside on the shore are also taken into account. We then identify the organisms by using Google Lens and also sources obtained from the internet.
Before we did aquatic insect sampling, we took 3 readings to get the average for river water speed. First reading we got 2.78 seconds, second reading we got 2.54 seconds, and third reading we got 2.42 seconds. This is the result that we got from thermoprodigital hygrometer thermometer. We got temperature and pH result from aquatic multiparameter. This is the result that we got from luxmeter. The first aquatic insect from our sampling is greasy-back shrimp. It is from phylum Arthropoda and fall under subphylum Crustacea. Its family is Pineidae and it fall under order Decapoda. Total species found is 1. Second aquatic insect is cockroach from phylum Arthropoda and fall under order Blatodia. It comes from family Blatidae and total species found is 2. Third aquatic insect is stoneflies from phylum Arthropoda and fall under Plecoptera. It comes from family Perlidae and total species found is 2. The fourth aquatic insect is freshwater worm from phylum Annelida and it falls under order Lumbriculida. It comes from family Lumbriculidae. Total species found is 1. The fifth one is lava cranefly from Arthropoda and it falls under order Diptera. It comes from family Tipulidae and total species found is 2. Next is Caridian shrimp which from phylum Arthropoda and subphylum Crustacea. It falls under order Decapoda and it comes from family Caridia. Total species found is 5. The seventh one is carp-tailed dragonfly from phylum Arthropoda and it falls under order Odonata. It comes from family Gomphidae and total species found is 1. Eighth one is spring stoneflies from phylum Arthropoda and it falls under order Plecoptera. It comes from family Nemoridae and total species found is 1. Next is gobyfish or gobis that comes from phylum Chordata and it falls under order Prustiformes. It comes from family Gobida and total species found is 1. We also found half-big fish from phylum Chordata and it falls under order Beloniformes. It comes from family Hemiphamphidae and total species found is 1. The last one we found was caddisflies from phylum Arthropoda and it falls under order Trichoptera. It comes from family Rheacophilidae and total species found is 40. The first invertebrate we found during sampling at Pantai Penyambung was sea cucumber from phylum Echinodermata and class Holothuroidea. It falls under order Dendrochirotida. It comes from family Holoturidae and total species found is 1. Second one is sea slug from phylum Mollusca and class Gastropoda. It falls under order Nudibranchia and it comes from family Onchididae. Total species found is 1. The third one is furrowed crab from phylum Arthropoda in class Malacostraca. It falls under order Decapoda and it comes from family Xanthidae. Total species found is 2. Fourth one is banded Dimurex from phylum Mollusca in class Gastropoda. It falls under order Neogastropoda and family Murichidae. Total species found is 1. Fifth one is Melon conch from phylum Mollusca and class Gastropoda. It falls under order Neogastropoda and family Melongonidae. Total species found is 2. Sixth one is rigid pen shell from phylum Mollusca and class Bivalvia. It falls under order Pterida and family Pinidae. Total species found is 1. Next is auger shell from phylum Mollusca and class Gastropoda. It falls under order Neogastropoda and family Terebridae. Total species found is 1. 8 is spider moon snail from phylum Mollusca and class Gastropoda. It falls under order Lithorinimorpha and family Naticidae. Total species found is 1. Next is black cockle from phylum Mollusca and class Bivalvia. It falls under order Archida and family Archidae. Total species found is 1. Next, we found hermit crab from phylum Arthropoda and subphylum Crustacea. It falls under order Decapoda and family Paguroidea. Total species found is 2. The last one, we found sandworm from phylum Annelida and class Polychaeta. It falls under order Philodocida and family Neredidae. Total species found is 1. Invertebrates we found from sampling at Pulau Batu Gajah were these three, namely hard-stony coral from phylum Nidaria and class Anthozoa. It falls under subclass Hexacoralia and order Scleractinia. Each of these three, we just found 1. These three are also hard or stony coral from phylum Nidaria and class Anthozoa. We just found 1 species for each of them. These three are also hard or stony coral from phylum Nidaria and class Anthozoa. We just found 1 species for each of them. These three are also hard or stony coral from phylum Nidaria and class Anthozoa. We just found 1 species for each of them. Next, we found flower crab from phylum Arthropoda and class Malacostraca. It falls under order Decapoda and family Portunidae. Total species found is 1. We also found sand dollar from phylum Echinodermata and class Echinoidea. It falls under order Clypestrida and family Echinarachnidae. Total species found is 1. Next, we found bristleworm from phylum Annelida and class Polychaeta. It falls under order Spionida and family Spionidae. Total species found is 1. Next is hard clam from phylum Mollusca and class Bivalvia. It falls under order Venerida and family Veneridae. Total species found is 1. The last one we found is sea snail or nereids from phylum Mollusca and class Gastropoda. It falls under order Cycloneritida and family Neritidae. Total species found is 1.
5.0 discussion. There are 3 activities that we have done. The first activity is aquatic insect and invertebrate sampling and identification in river or waterfall and its surrounding. And the second activity is observing and identifying the marine invertebrates that reside at the shore of the Pantai Penyambung, Mersin Johor. And the last activity is observing and identifying marine invertebrates at Anak Kacang Island. For the first activity, part A, in this activity, it focuses on the on aquatic insect and invertebrate sampling methods and identification in a river or waterfall and its surrounding at the Hutan Simpan Gunung Arang. Our group was set in an area with rocky, shallow, and shady at the middle part of the river. We had to learn how to use super net by shaking rocks method using aquatic net and how to measure the abarotic factor like humidity, light intensity, river pH level, speed of the river current, and the soil temperature. Since it was raining during the sampling period, it affected the sampling process where aquatic insect and invertebrates are a bit difficult to be observed. Abarotic factors, humidity and temperature are measured by using the thermoprobe digital hygrometer thermometer. The humidity reading is taken 3 times to get an average, which is 97 RH relative humidity, while for temperature value is 26.4 degrees Celsius. The light intensity may influence the animal activity and behavior because the population measurement of the fauna can become distorted as behavior changes in response to light. The equipment used in this activity is H197500 portable luxmeter with the value recorded is 0.07 lux. Next, the pH level is measured by using the aquaric multibarometer, which is 6.71 pH that can be considered as acidic for our sampling area. Lastly, the water velocity is measured by using a ping pong ball and a timer, and the average velocity taken is 2.558. meter per second. For Part B, it focuses on the identification and determination of the aquatic insect and invertebrates at the Center for Marine Ecosystem Research, Ekomar, UKM. The insect that we collected were from phylum Arthropoda, and for the insect in larvae stage, mostly we found it by looking at the rock surface and underneath it. One of our sample, the greasy bag shrimp, was found in our area of our sampling. The shrimp is from subphylum Crustacea and falls under order Decapoda. The shrimps were found due to our sampling area being considered as a place with low water current flows. Next, we found caddis flies that fall under class Insecta with order Trichoptera. The head of the larva seems like a box type of shape. In addition, it also can be a good bioindicator for the quality of the freshwater ecosystem in the river. It has the highest total species found in the sampling area, which is 40 in total.
Next, I will continue the discussion about the activity we do for the second activity at Ekomar. We observe and identify the marine invertebrate of the Pantai Penyambung, Mersing Johor. In this activity, it focuses on aquatic invertebrate identification in the beach and its surrounding at Pantai Penyambung. The fieldwork continued after our lunch break, and we went to Pantai Penyambung to collect invertebrates sample along the muddy area at the seashore. We observe and collect an invertebrate sample at the seashore of Pantai Penyambung. We arrived at the site and started to observe organisms that we could find in that area. We observed the space between two rocks, puddle and mud. Most of the invertebrates we caught by hand and we observed before we release them back to the beach. We also used to take picture of the invertebrates for next activity, which is the identification of them. For this activity, we moved in group to observe the beach area. The weather for that day was quite windy and dim. This is how we collect and observe the invertebrate at Pantai Penyambung. This is how we collect and observe the invertebrate at Pantai Penyambung.
Moving on to the third activity, which observing and identifying marine invertebrates at Pulau Batu Gajah. For this activity, we were assigned to snorkel around the island for the observation and identification of marine invertebrates. Before performing the snorkeling activity, we were supplied with snorkeling equipment and instructed on how to use it. Since this fieldwork was done during monsoon season, we were not able to observe any marine invertebrates on seabed as visibility in the murky seawater was limited. However, we have found in total 17 species of marine invertebrates around the beach of Pulau Batu Gajah. We have found 12 coral species from the phylum of Cnidaria that belong to the class of Anthozoa. We also found a sand dollar from the phylum of Echinodermata that belongs to the class of Echinoidea. After that, we found a flower crab from the phylum of Arthropoda that belongs to the class of Malacostraca. Other than that, we have found a breastile worm from the phylum of Annelida that belongs to the class of Polychaeta. And finally, we have found two types of marine invertebrates from the phylum of Mollusca that each belong to the classes of Bivalvia and Gastropoda. Based on the result, corals that belong to the phylum of Cnidaria are the most species of marine invertebrates that we have found at Pulau Batu Gajah. These corals are important for the protection of other small marine invertebrates to hide and as a shelter for them and live as colonies. As for the conclusion, the sampling method of aquatic insect and invertebrates by using the sabon net, aquatic net, and stone shaking technique has achieved better understanding and conducted in a great performance. There are several abiotic factors that needs to be identified and considered throughout our finding, in which these factors have very significant impact on the survival and the reproduction of species in the ecosystem. For instance, humidity, temperature, pH level, light intensity, and water velocity. The parameters of abiotic factor are recorded by using the ThermoPro digital hygrometer thermometer, AquaRead multiparameter, high 97,500 potable lux meter, a ping pong ball and timer. The abiotic factor are important to have strong influence on area distribution of the species. While conducting the finding on 3 different study areas, the aquatic insect and invertebrates are collected for further identification. The important characteristics of each aquatic insect and invertebrates are identified carefully. Hence, some of the aquatic invertebrates such as echinoderm, annelids, mollusks, and arthropods have been well observed with the group members through their unique figures. Overall, the study of invertebrates in this fieldwork has proved their existence as the bioindicators to the system and the keystone species that help to define an ecosystem.